Good morning, students. Today we'll study the major histocompatibility complex, that is MHC. It is a very important part in immunology as it represents cluster of genes arranged within a continuous stretch of DNA on chromosome number 6 in humans. It is also known as Human HLA complex, that is Human Leucocyte antigen. It is responsible for providing cell surface glycoprotein receptor on the cells which are responsible for presenting the antigen to T cell. MHC basically is divided into three classes: MHC class 1, MHC class 2 and MHC class 3. MHC class 1 has three regions A, C and B, which leads to the production of HLA-AA, HLA-B, HLA-C, whereas MHC class 2 has three regions, namely DP, DQ and DR, which are responsible for producing gene products as DP alpha beta, DQ alpha beta, DR alpha beta, whereas MHC class 3 is having regions as C4, C2 and BF, which leads to the production of gene products like C proteins in the complementary system and TNF alpha beta, that is tuber necrotic factor. MHC class 1. MHC class 1 consists of two parts, alpha and beta 2 microglobulin. Alpha chain is divided into three parts: alpha 1, alpha 2 and alpha 3. Alpha 3 is attached with the membrane with the help of transmembrane segment with cytoplasmic tail, whereas beta 2 microglobulin is not attached with the membrane. Alpha 3 is an important site which is responsible for binding with CD8 lymphocyte, whereas the groove which is formed by alpha 1 and alpha 2 is responsible for binding with the peptide. MHC class 1 molecule enables the body to recognize infected cells and tumor cells and destroy them with cytotoxic T lymphocytes. Alpha chain, as I've told you, is made up of three domains: alpha 1, alpha 2, alpha 3. Each domain consists of 90 amino acid. Alpha 3 continues with transmembrane domain of 25 amino acid and cytoplasmic tail of 30 amino acid. The groove which is formed by alpha 1 and alpha 2 forms peptide binding cleft, which can bind with antigen of 8 to 10 amino acid long. Beta 2 microglobulin is small. It is coded by 15th chromosome on the human body. It does not contain any transmembrane region and it is non-covalently linked with alpha chain. The main function of MHC class 1 are: MHC class 1 binds to the peptide antigen and presents it to the CD8 T cells, that is T killer cells or cytotoxic cells. CD8 T cells are very specific to MHC CLASS1 antigen. MHC CLASS1 binds endogenous antigen and are found on all nucleated cells. The expression of MHC molecule is increased by cytokines produced during both innate and adaptive immune responses. Cytokines like interferon alpha, interferon beta, interferon gamma, and tumor necrotic factor. Now, what is the role of MHC molecule in adaptive immune responses? MHC molecule enable T lymphocytes to recognize epitopes of the antigen and discriminate self from non-self. Very important, B cell receptors directly bind with epitopes present on the antigen, but T cell receptors cannot because they need processed antigen. Both endogenous as well as exogenous antigens are processed inside the cell, and the peptides, they are presented on MHC molecules to the T lymphocytes. Endogenous antigens include the viral proteins, intracellular bacteria, antigen presenting cells, tumor antigens, self-peptides from the host cellular protein. This is the process, how it takes place. Now, number one is the endogenous antigen. It enters into a proteasome. Proteasome breaks the endogenous antigen into large number of peptides. These peptides enter into the endoplasmic reticulum through TAP, transporter protein. In the endoplasmic reticulum, we have MHC class 1. The broken peptides bind with the MHC class 1 and enter into the Golgi complex. From the Golgi complex, the MHC class 1 along with the peptide enters into the vesicle. Through the vesicle, it comes out on the surface of the cell and get attached. Now here it binds with CD8 molecule and is responsible for lysis. Now coming to MHC class 2 molecule. MHC class 2 molecule, again they are heterodimers having two chains, alpha and beta. Alpha has two parts, alpha 1 and alpha 2. Beta again has two parts, beta 1 and beta 2. Both these chains, they are having transmembrane sequence as well as the cytoplasmic tail. Now, the groove which is formed by the alpha 1 and beta 1 are responsible for binding with the peptide. Here, larger sequence of peptide can bind. Maybe up to 15 or 20 amino acid long residue can bind with MHC class 2 molecule. Now, MHC class 2 molecule is found on antigen presenting cells. Basically, dendritic cells, mononuclear phagocytes, endothelial cells, thymic epithelial cells, as well as the B cells. These cells, they are very important and they present the antigen to the lymphocytes. Now see here in the diagram, suppose the bacteria enters by endocytosis, it forms a phagosome. This phagosome, it binds with the lysosome to form autophagosome. The enzymes from the lysosome, they will disintegrate the pathogen into large number of peptides. Now, endoplasmic reticulum, again here also it plays a very important role for the synthesis of MHC class 2 molecule. This MHC class 2 molecule has invariant chain as II chain. It is attached with the MHC class 2 molecule. It enters into the Golgi complex. In the Golgi complex, invariant chain is broken down. Clip is formed which is bound with the MHC class 2 molecule. Now through vesicle it is fused with the polyphagosome, and here the clip is removed, and instead of that the peptide is attached with the MHC class 2 molecule, and through the exocytosis the MHC class 2 molecule gets attached with the surface of the antigen presenting cell. Now this antigen presenting cell has the MHC class 2 with bound peptide epitope. Now see here, this is the difference between the two MHC class 1 and MHC class 2. In MHC class 1, endogenous antigen is processed with the help of proteasome, whereas in MHC class 2, the antigen is processed with the help of lysosome. Now this is basically the difference between the MHC class 1 and 2. Class 1 is having alpha 1, alpha 2, alpha 3, whereas class 2 is having alpha 1 and alpha 2. Class 1 is having beta 2 molecular globulin, whereas class 2 is having beta 1 and beta 2. Here very important is the peptide binding cleft. In MHC class 1 is formed by alpha 1 and alpha 2, whereas in MHC class 2 is formed by alpha 1 and beta 1. Now in MHC class 1, CD8 binds at alpha 3, whereas in the case of class 2 molecule, MHC class 2 molecule, CD4 binds at beta 2. Now when we look at the difference, the structure difference is clear. Types of APC is also clear, because in the case of MHC class 1 only all the nucleated cells they will have the MHC class 1, whereas in the case of MHC class 2 only antigen presenting cell will have the class 2 glycoproteins. Now in MHC class 1, cytotoxic T lymphocytes they will be presented with the antigen, whereas in class 2 helper T cell lymphocytes will be presented. Co-receptor responsible, in the case of class 1 it is CD8, there it is CD4+. Then cytosolic proteasome is responsible in the case of MHC class 1, whereas lysosomal proteases and endosomal proteases they are responsible for breakdown of the peptide in class MHC class 2. Now the end result in MHC class 1 is presentation of foreign intracellular antigen to CD8 for destruction, and for MHC class 2 presentation of foreign extracellular antigen induces antibody production and attracts the immune cells in the area of infection. So this is all. Thank you.